Well, good morning, ladies and gentlemen. As the sign says, the past is the sculpture of the future. Not just of cookbooks, not just of culture and tradition, not just of the University of Otago, as you've seen earlier, not just also of the fact that old habits die hard, but in fact the past is a biological force that changes our future and the patterns of the future, and that you'll see a little bit about that in my talk. To begin with, I want to illustrate something to you. Some of you will remember the 1980s. The 1980s changed us in a number of different ways. It changed us in the things that we do. It changed our cookbooks, if you heard. But also it changed our phones, our entertainment, all sorts of things. For me, it changed me entirely. This is me in the 1980s, and I made a choice in the 1980s when I finished school to come to the University of Otago and study medicine. And that choice has changed my life entirely. I did a medical degree, I met Jim, I did research there. I went on to work in Melbourne and now run a research lab in Australia. And it's really fascinating to think that all of those choices that I made as a high school student here in Wellington influence me today. But I mean, I say that's just in my head. I'm just nostalgic of wandering through the streets of Wellington. But actually there's biology behind it as well. And that's what our lab focuses on. Because if you imagine in the 1980s, there were a number of interesting studies. One of which is the most important for that is the Da-Qing study done in China, where they took individuals in the 1980s, as you would do, and say, some of them, we would apply diet and exercise, aerobics, and the kind of diets that they ate in the 1980s, and the rest of them received their status quo, the standard management of people with pre-diabetes at that time. And then they applied this strategy for 6 years, dieting and lifestyle. And then at the end of that study, they sent them back to their communities. They told them, do whatever you like, this study is over. Importantly, this study during the intervention period, and these plots show the white line being the standard therapy and the red line being the intensification, showed a significant reduction in the incidence of diabetes. But what was really remarkable that over the next 23 years, those people who found aerobics and dieting in the 1980s were still ahead of the game. They had never caught up. They had a substantial legacy so that they were, very few of them developed diabetes overall. Equally, when you look at their mortality, of course, obesity, diabetes, mortality comes later after that. You can see that those people who changed their ways in the 1980s, even though they are no longer doing those things anymore, they're outside of that trial situation, they still are retaining the benefits. 30 years later, 3 decades later, they are still retaining the benefits of a good diet, an increased level of physical activity, such that now, essentially, there's a quarter reduction of being dead as a result of what they did in the 1980s. But it's not just in diabetes as well. There are a number of studies that show this. And this is another example, the West of Scotland study, where they put individuals in the general population in primary care on a statin to lower their cholesterol levels. And of course, you know lowering cholesterol is beneficial for you. And the trial lasted for 5 years, showing those benefits. But what remarkably happened after the end of the trial is that they couldn't keep taking the drug. They were told, go back and be managed in the real world. And they were managed in the real world. But because they were in that active treatment arm, receiving that statin, pravastatin, now 20-something years later, they are still seeing the effects of once having lowered their cholesterol levels. Even if it's not continued, the benefit persists over a long period of time. And it reminds me of the famous cartoon that says, hey, I remember you from the usual control group as they reach heaven. And these, it's exactly like this in trials. If you're actually in an active treatment arm and the drug works, actually you'll get a benefit. But interestingly, in these studies, the benefit persists almost until the day that you die. And one wonders if you imagine, for example, what happened in New Zealand when reference pricing came in and cholesterol levels rose, what kind of legacy that has created for the future as well. My research is focused on how on earth this kind of legacy can come about. And we have termed this quite simply metabolic karma. A very simple term because everyone knows about karma. It means intentions and actions have consequences. But if you imagine with respect to metabolism or diet and lifestyle, the same sort of thing exists, whereby your intentions and actions, the control that you achieve through those things, and importantly, the suitability of those actions for you as an individual might somehow represent a causal force that change you in a way to deliver future outcomes. And that, if you like, is metabolic karma. But how do you think it works? How could something carry forward? You can imagine your memory. I remember what happened in the 1980s. Memory might work that way. But how can cells remember what has happened in the past? Now, there are a number of different ways that this can come about. One of which is something called lead time, or in other words, a head start. And this is illustrated here. So if you imagine two yachts, a good New Zealand analogy, two yachts starting at about the same time, but one has a slightly greater wind at the beginning, maybe running at 9% instead of 7%. So it advances just that little bit faster or a little bit further. And then the wind changes that everyone gets the same wind subsequent to the trial. Which one finishes first? The one that had the head start towards the finish line. And so whether the reaching of the tipping point is the progression of a disease process or a shortening of telomeres or some other marker of disease, if you get a head start at the beginning, even if you carry on very well when you realize your diet and lifestyle was important, the loss that you have at the beginning can eventually mean that you finish first or last, as the case may be. An example of this is a trial done in patients with type 1 diabetes, where they, for around about 6 or 7 years, lowered their glucose levels from relatively high levels down to what was then an intensive level at around about 7%. And what you can see is that during the trial it had no effect because no one really got into trouble. But in the long-term legacy, when you're looking at the incidence of developing kidney problems, those people who had the status quo, you can see developing problems at around about that 10 to 15 year mark and then increasing beyond that, whereas those people who had that intensive control for that period of time got pushed back. Exactly the same number of years they were in the trial, they've pushed back their development of complications. So the lasting legacy now, decades later, of the things that they did in the 1980s is readily apparent. The other reason that this might work is something called the Humpty Dumpty paradigm, and many of you will be absolutely familiar with this, because sometimes all the king's physicians and nutritionists can't put Humpty back together again. There are some cells in your body that are essentially can't be regrown. They are what's called post-mitotic, or can't grow again. Things like the beta cells in your pancreas that make insulin, the heart cells, the podocytes, and even the neurons. Once they are lost, they're gone. And so even if interventions come in to say, hey, I might protect the heart or the brain or the kidney, if these cells are not there, they're not going to come back. And that is how legacy might work under some circumstances. Another way is accumulated modifications. And this was the topic essentially of my PhD at the University of Melbourne and subsequent research going across, to look at how it is that, for example, the sugar from a Louise cake that might happen today could influence my future overall. And one of the reasons is that we are not simply just proteins. Those proteins can be modified by sugar. And so if you cook, for example, your pork with some with some honey, you get that lovely glaze on the surface and it becomes crunchy and tasty. And the same sort of thing happens in bread when you put it in that toaster, that lovely toasted taste on top. So essentially, this is your knee ground up. And you can see the same protein all the way across, the same genetic code, but a vastly different protein altogether. It's become yellower, it's become more crunchy, it's become, as you get old, more crusty. And exactly the same process. This is chemical modification, where the sugars in your bloodstream circulating at 37 degrees are slowly glazing, if you like, your tissues. And particularly those things that are long-lasting, the lens proteins in your eye, the basement proteins around your arteries, and your cartilage in your knee, which doesn't turn over except for every 30 years or so. You are accumulating the modifications. And of course, if you have a period in which those accumulation is faster, you have a head start towards arthritis or other changes as well. And trying to get rid of some of these modifications is something that we're working on at the moment. Of course, there are other changes as well that lead to metabolic karma. The most obvious one are compositional changes. If you like, the scars that we have. We all have famous scar stories. You know, you've all probably got one scar on your hand where you cut yourself. You remember what you did, and it carries forward, and you go, this is not the same skin that I used to have. This is a scar tissue. But other changes as well, and the most obvious one is obesity. So that when you gain weight and when you gain structure, you've changed your composition. You've got a different beast that you're dealing with. And as a result, you can expect different outcomes altogether. And some of this relates to simple physiology, but some of this also means rewiring. And another famous story, of course, is the story of Goldilocks and the Three Bears. You might recognize this because the big bear sits in a big chair, has a big bed, and eats a big bowl of porridge. Why? Because he's a big bear. And so it's entirely appropriate for that situation. And so what we find is that people who gain weight, at least after their adolescence, suddenly their brain says, you're a big bear. You should be receiving a big bowl of porridge. You're not like Goldilocks, you're a big bear now. You should be accepting that. And so you feel uncomfortable in the little chair. You feel uncomfortable with the little bowl of porridge. And that rewiring is absolutely real because you're dealing with something else entirely different. But finally, I want to talk a little bit about a really exciting new emerging science, and that is epigenetic reprogramming. And some of you will have heard of this, but if you haven't, here's a really exciting crash course in it. So if you imagine that genes are the words that tell you what's going on to make a protein or whatever, epigenetics essentially doesn't change the words, doesn't simply highlight one or scrub another one out, but actually allows you to open a page at a particular time and address a particular word in a particular setting in a particular way. So essentially it instructs the use of the words rather than being the words themselves. And there are a number of different changes which are termed epigenetic. So imagine this is your DNA strand looping up on the far side. You can actually chemically modify that DNA. And that chemical modification, known as DNA methylation, can actually mean that that piece of DNA is silenced. So you can have a bad gene that's turned off and never know about it. You can have a good gene that's turned off and never benefit from it. You can have insulin gene in your foot, but of course your foot doesn't make insulin because it's turned off. So differentiation between cells, between settings can be done through epigenetics. You can get modifications, and these tiny little blue strands up there, RNA strands, can bind over areas and actually, instead of just making proteins, actually control whether that gene is red or not red. Another change, in particular one that we're interested in, is the changes in around these little proteins called histones, in which the DNA wraps and wraps and wraps themselves around. And that wrapping determines whether the gene is available or not available for transcription or ability to be used. And what determines this is a whole new level of code called the histone code. And so essentially, if you imagine there's the tail of histone 3, and that tail has got signals on it. And if you have right down at the end, there's a little K there representing a lysine. And if that's methylated, that's a signal that that gene should be turned on. Whereas if histone 3 is methylated at K9, therefore that gene should be turned off. And so you can have a whole genetic code, but on top of that, there's a whole regulatory code that says whether a gene is open for business or closed and no one wants it at all. And this can work in a number of different ways. And you don't need to look at the names. These are all genes that we've been working with. All of these are silenced in healthy individuals because of essentially epigenetic instruction. But if you then pour onto these cells high levels of sugar, high levels of glucose, this generates reactive intermediates through the metabolism of that sugar, not the sugar itself, but its metabolism, that triggers new epigenetic modifications, that turns these off genes into the on switch. You see all of these genes now turned on through different epigenetic modifications, and you see inflammation as a result. But most interestingly, what happens if you take the glucose away? You can come back 10 days, you can come back 2 weeks later. These changes persist. The cells remember the fact that you poured glucose on them 2 weeks ago. Glucose is not there, but the cells have a memory too. And the memory is partly through these epigenetic modifications that persist. And so when you look at inflammatory changes now, weeks after sugar exposure, you can see that they're still going. Sugar's not, but the changes are still there. And of course, how does this reflect on, for example, our weekend-centric behaviour, where our sugars go up on the weekend when we overindulge and we're more modest through the weekdays? We've done that experiment recently in animals where we've essentially raised their sugar levels just for one day on the weekend for 8 hours and looked at their development of diabetic complications. Not only do they develop complications, but they develop just as many complications as animals that have got consistently high glucose for the whole period of time. So those transient spikes from those high GI foods, those overindulgences, they add up. They are epigenetic modifiers. And of course, these aren't the only ones. Of course, everything modifies us. Every choice that we have modifies us. That makes it exciting. But also, the fact that some of these changes can be erased is also more exciting as well. So that we are the dynamic flux of our past, choices, good and bad, and the accumulation of that is essentially metabolic karma. For those of interested in cancer, this also works, and a number of the cancers that we know about turn out to be beginning not through some genetic aberration, but actually through an epigenetic origin, whereby some genes, oncogenes that are cancer-promoting, that are normally silenced in young people, and of course tumour suppressor genes that are liberally active in young people, mean that cancers are very uncommon in younger people. But with changes through time, through diet, through aging, through exposure to epigenetic modifiers like plasticizers and other kinds of things, you can get changes such that epigenetic liberation now comes to the cancer-promoting genes, whereas aberrant silencing now comes on the suppressors. And many cancers, particularly in childhood, but also in adulthood, begin with these first epigenetic changes. And of course, when you open a gene up, also it's sitting there in the breeze waiting to get damaged, and that facilitates mutation and from there. So epigenetic origins are really important for explaining not only diabetes and cardiovascular disease, but also the origins of cancer as well. So in summary, really what you want to know about epigenetics is very simple, that your diet and lifestyle, without changing the gene sequence, can actually change the structure of the chromosomes to actually shape essentially the future as a kind of memory overall. And this overarching level of instruction perhaps is more extensive than genetic regulation. Another kind of example, and I want to mention this here because Ian Pryor essentially was a doyen of medicine and indeed epidemiological research here in New Zealand. And really, he was one of the first ones to describe what he described as the metabolic maladies in New Zealand Maoris, published in 1964 as a lead-on for him from his 1962 work with the Ministry of Health. Essentially, he describes that there was an excess of risk in Maori New Zealanders, in which he argued that genetic factors were likely to be important. But it turns out that genetics only explains a very small proportion of this variability. And in fact, there may be other layers overall, not genes, as you heard of before, not diet, not genes, but what's in between, helping the diet influence the genes, helping the genes influence the diet, actually is the metabolic karma of epigenetics. And another example, obviously, is somewhere like Nauru, where the genes haven't changed in that many generations. But obviously the diet and lifestyle has. And so maybe epigenetics essentially is changing or programming the predisposition. And many of you will know the history of Nauru, where after the Japanese occupation, most of the island islanders were removed off to Micronesia, and those people who survived the work camps actually were then repatriated and then exposed to the diet of their future. So essentially, you've not only got an adaptation, not only got a survivor bias from those as survivors, but those who have got the adapted survivor, hold on to my calories adaptations, are now able to not only suffer the effects of their diet, but critically pass those effects on to their children. Not through changed genes, because epigenetic changes are heritable. Because the epigenome is not only influenced by your diet, by your lifestyle. It not only has a direct effect on genes, but your genes themselves, for example, in Wilms tumor, can affect the epigenome and therefore the chances of tumor. But for example, studies in Sweden, the Overkalix study, looking at individuals, their grandfather's diet determined their risk of developing obesity and cardiovascular disease. When you look at survivors of the Dutch winter famine, again, key epigenetic changes are not only seen in the children, but their children's children, because of the ability of adaptations to be transported through multiple generations. And it's really exciting to know that at the University of Otago, they've now established a new group on epigenetics, and one of those is on the transgenerational effects of epigenetics, and whether the historical trauma that exists in many countries and in many people and in many cultures actually has a payback in terms of health, in terms of dietary choices, in terms of nutrition. So, in summary, ladies and gentlemen, thank you so much for being able to talk a little bit of science to you today and tell you about my stories in getting here. I want you to take away that memories are actually not just about the past. They're not about going back to tradition or whatever. Memories are a way of actually transforming the future because they have a real, not just emotional, not just nostalgia, but actually a real biological effect. And that our choices change our biology as much as they change our waistline. At the same time, there's a real opportunity to show some intent and take some actions. And as you'll hear later at the symposium, there are many important ways that we can do that. And the reason that we do that is not to shape now, but to shape our future. Because there's a real opportunity that improving our diet, reducing our portion sizes, reducing enrichment, doing all of those things as well, can cast an important legacy of good karma for our own health and for the health of our children into the future. Equally, by not making those changes, by delaying, by showing inertia, by going, oh, it's a bit too hard, or we'll fix it next year, it catches up with us. And as you've seen from these studies, even brief periods of poor control can have a long-lasting legacy of bad karma into the future. And with that, I'll leave it there. Thank you.
So do we have any questions? Professor Thomas, yes.
Thank you for that. I find this whole,
Lisa.
This whole epigenetics thing to be quite exciting, but also confusing. Can you turn those genes off again? And in those legacy effects that you're seeing in the Dutching studies, would that be an effect of unmethylating those genes?
Yep. So the question is, what would happen if you took away memories? would you be much better? Would you be much worse off? And just to give you an example, you know this one, right? You know, this is Dory, okay? And Dory has no memories, right? Is Dory disabled or is Dory enabled? In fact, I listen to this in the car all the time. This Hank the octopus says, no memories, no worries, you know? And that may be the case for some genes, and it may be that sometimes memories are actually an important driver. Now, how can we selectively change particular genes to turn them off and turn them on? That is the real exciting thing. We don't know. We know that they're involved, but we really don't have the tools to say we can selectively modify things yet. Now, we're working at the moment on developing what's called, a little complicated, called artificial transcription factors, by putting a protein that actually involves DNA methylation, attaching it to a code that recognizes a particular gene, so that when it walks through the genome and actually it sits on a gene that's bad and then turns it off epigenetically. So some of those kind of things are being developed, but currently we have no good way of saying, yes, we can erase one gene or fix another, because the complexity that exists within the epigenome of turning on and turning off is not only complex for the understanding, but it's also complex for the manipulation at the moment. Yell it out. Or...
Thank you very much for the talk. I'm really fascinated by this whole relative proportion of environmental risk, genetic risk, now in epigenetic at the same time. And my question really relates to the epigenetics. Do you see a lot of individual variation in to what extent genes are methylated, for example? You know, given the same exposure, are all individuals going to react the same way? Or does the underlying genetics influence the epigenetics, if you like?
You're absolutely right. The complexity of all of this is mind-blowing. But overall, it's very clear that for some people, their genes, for example, have a major effect on how their epigenetics works. And a classic example of this is the Prader-Willi syndrome, the most common cause of genetic childhood obesity, where a genetic change influences the epigenetic silencing of certain targets and results in hyperphagia and other issues. For example, in Wilms tumor, a common tumor of children, where a genetic abnormality leads to aberrant silencing of a particular tumor promoter thing. So it can work that way, but it can also work the other way. So the interactions between genes, environment, and epigenome is complex, still not well understood. But the fact is that there's now this multiple layer between them to allow, for example, the food that we eat, the exercise that we take, not only to change our fitness or our fatness, but also at a fundamental level to change how our DNA works. And that's really exciting if you want to say, you know, I want to know what my diet is doing. I want to know what my lifestyle is doing. In the future, I suspect we'll be able to see how it changes how our DNA is on or off, or maybe there's an intermediate switch somewhere in between. I don't know, but it's really exciting.
Just one more, Kirsten.
Thanks, Merlin. That was really great. I'm just aware of the fact that the Darjeeling study was done in adults. And
Yes.
so just thinking about this legacy effect, has there been any work looking at different ages that this may impact? Because clearly pregnancy, young children are important for us to think about.
Well, absolutely. There are times in our life that we turn over our epigenome. We, if you like, we erase and start again. One of those times is during conception, but there are other times early in adulthood that you can change early in development, that you can change your ways. And in fact, a lot of work has looked upon the effect of in utero and early development on epigenetic imprinting, particularly in indigenous populations. And we've worked, for example, in Australia on that, showing that some of the effects of intrauterine growth retardation, or being born a small baby, not being able to develop as much, transforms not the genes, they haven't changed, but actually the programming of those genes, so that in the future those younger individuals are more prone or more susceptible to certain diseases, including obesity and hypertension. And one classic example of this is the Dutch winter famine survivors, whereby those individuals that were conceived and born during the Nazi occupation in World War II, they have a long-lasting legacy that when exposed to diet, they have got much higher rates of obesity, hypertension, and some of that relates to their epigenome, and just one of those changes, they can see that they've got more silencing of a particular gene, more methylation of a particular gene. So the answer is yes, absolutely. Early development is critical, not for just developing brain and developing kidneys and developing other things, but actually programming a healthy future.
Well, thank you. On behalf of the committee and everyone, here's the gift for you. Thank you very much.